Start? Hey, thanks for inviting me. It's a real pleasure to be here. So one of the very first talks I saw as a PhD student was from Gonzalo Abacasis. He said, the job of the statistical geneticist is to step into the light and shine darkness. And so I try not to give all my talks like that, but this is one of those talks. So genetic and environmental variation underlie the phenotypes that we're interested in. Behavioral phenotypes, disease risk, metabolite levels, and this sort of model has been used to develop what is quite a prolific approach to identify the genetic basis of human disease, GWAS, and for hundreds and hundreds of traits, sometimes with studies reaching the millions of individuals. We found genetic variation that is correlated, associated with complex traits across the genome. And the community of statistical and quantitative geneticists has thought about the resultant data and developed a bunch of new tools, polygenic estimators, that are not necessarily interested in whether an individual variant is associated with a complex trait or not, but is try to do other clever things with that data, to estimate genetic correlation between pairs of traits, to identify causal relationships between pairs of traits for things like Mendelian randomization, to estimate heritabilities, to figure out the cell types and developmental periods in which the genetic variants associated with a trait are acting, and to determine the effects of selection and evolution on these traits. And for nearly all of these methods and all of these papers, there's probably a new one coming out every week. There's a section of those papers that they establish that the method works. And the way that they establish that the method works is through empirical simulation. They simulate some trait. I think of it as more like half simulations. It's real genotype data. They simulate some phenotypes. They apply the polygenic estimator, and they show that they're getting an unbiased estimate of the thing that they're after. These simulations typically almost always assume additivity and random mating. What we're thinking about, I should say this is the work of Richard Border, a former postdoc in my lab who just started his faculty position at Carnegie Mellon University, and Jamie Matthews, who's just finished her PhD and is looking for postdocs, if anybody is looking for a very good postdoc. So what we were thinking about is intergenerational dynamics, assortative mating, vertical transmission, and how what happens in one generation influences the genome and the environment of the next generation, and whether these types of intergenerational dynamics are going to influence and bias the polygenic estimators. And to accomplish this, Richard developed this software platform called XFTSim. If you're from the field of population genetics, you probably use things like PopSim or SLIM. This is meant to be that tool, but for statistical and quantitative genetics. And so instead of just an additive model with random mating, it can do much more complicated forms of simulation across multiple generations, but for the last 5 generations. Not like SLIM, where you're looking hundreds of generations back or tens of generations back. And it can look at vertical transmission and gene-environment interaction and nonlinear effects and forms of population structure. And this software is sort of the basis for a lot of the stuff that I'll be talking about. And it's available, and the paper describing it is under revision right now. So let's talk about an example of a polygenic estimator and how this type of intergenerational dynamics can impact it. Let's talk about genetic correlation. Genetic correlation, is a very widely used tool. I think probably one of the most impactful areas of the application was in psychiatric genetics, where there was, upon first application, quite some surprise in the sets of traits that were found to be genetically correlated, like between ADHD and schizophrenia, that were perhaps inconsistent with things like sibling relative risk. Now I'm going to switch and sort of define it more specifically. Okay, so if we think about a trait like height, And we think about an additive model for height. We have some G's and C's and A's and T's, and the ones on top increase your height, and the ones on the bottom decrease your height a little bit, and then there's some non-heritable factors. And we think about our trait Y as the sum of these things. So this is an additive model for height. We can think about another trait, which is education years, the number of years that you spend in school. And we can model it the same way. One thing we can look at is the effect correlation. That is really, truly, for the causal variants, they have an effect size, and if we look at the correlation of those effect sizes, that is probably what most of us think of when we hear the word genetic correlation. If I alter this variant in the person, they will both have a different height and they'll spend a different amount of time in school. And we will write that rho B, rho beta. The other type of correlation, which is different, is the correlation in what we call liability. You can think of it as like the perfect polygenic risk score. So if we sum all of those things together across the individuals, and then we take the correlation of that, we look at the correlation of polygenic risk scores. And under additivity and random mating, those two things are identical, but when we start breaking those assumptions, they're not. And so now, if we look at the papers that have proposed and used genetic correlation, I'm not calling anyone out specifically, they all do this. They interpret genetic correlations as effects that are causally impacting multiple traits. That is, there is a shared genetic basis for both of those diseases. And have gone on to make sort of a further sort of inference about the nature of diagnostics and all kinds of stuff on the basis of these genetic correlations, and they make this assumption. They make this assumption of random mating. Okay, you might feel like you won the lottery in finding your spouse, but you probably did not quite literally play the lottery to find your spouse, and we don't. Right? And so if we look across us spouses, we look across people who have kids together, they're correlated on a huge variety of traits. Tall people tend to mate with taller people. People who spend a lot of time in college tend to have kids with people who also spend a lot of time in college. And there's also what I'm going to talk about now, a cross-trait assortative mating. So it is also the case that if you look in populations around the world, people who are taller tend to have kids with people who spend more time in college, and vice versa. Okay. And so this is also true within psychiatric disorders. So people who have a diagnosis of anxiety are more likely to have a spouse with a diagnosis of schizophrenia, also within and between psychiatric disorders. Okay, and when we started this, what we did is we went to the UK Biobank and we identified all the spouse pairs that we could find, and we compared the spousal correlation between, I think, a couple dozen different traits and the genetic correlation estimator that comes back from running one of these tools. And we found this to be pretty remarkable, that we didn't need to pay for all of that genotyping if we wanted genetic correlation estimates. We could have just interviewed people and asked how correlated you were with your spouse for every single one of these traits. So this was a bit nerve-wracking, and we decided to think about it and figure out what was going on. I think the heart of the matter is really the long-range LD that is induced by the cross-trait assortative mating. So here's what's going on. If your even chromosomes, I'll put them in pink here, influence height, and your odd chromosomes, which are in yellow, have no impact on height. And your yellow chromosomes impact the number of years you stay in school, but have no impact on height, right? And sort of vice versa. The even chromosomes do not. And in a random mating population, there's going to be no LD, right? We recombine our chromosomes when we make our zygotes, and everything's fine. But as soon as you get this cross-trait assortative mating, where people who are taller are having kids with people with more years of education, then all of a sudden there is linkage between the causal variants on the even and odd chromosomes. And this breaks the assumptions of all of these polygenic estimators, which are based on LT and things like this. Okay? So we did a forward-time simulation using sort of cross-mate correlations and heritabilities that were reasonable, that were based on parameters that we were getting back from the UK Biobank, and we looked to see how these were influencing a lot of different polygenic estimators, in this case, a genetic correlation. And so this is what we found. We simulated a trait where truly the score correlation is 0. There's absolutely no effect of trait 1's genetics on trait 2's genetics and vice versa, completely independent. And then we looked across subsequent generations of assortative mating. The black line is the score correlation. So that is in some sense real. That is, if you build a if the population is assortatively mating, and you build a polygenic estimator or polygenic risk score for height, you will learn something about the number of years that person is staying in school. But it's different than embryo selection, which I think some people have been talking about through the conference, that will not be predictive of what happens through embryo selection, right? It's a different thing. And then there's the estimators themselves, which are that we use in practice. One is Hausmann-Easton and one is Remmel, and they're both biased. They're both substantially biased even beyond the score correlation. And so we did a more specific study within psychiatric disease, trying to ask the question about the ratio of what we see predicted based on the parameters we have observed from the cross-mate correlations and the heritabilities of these traits and the empirical correlations that come from these tools. And what we found was after 3 generations of assortative mating, that a large fraction of the genetic correlation between pairs of psychiatric traits could be fully explained, a large fraction of it could be explained by cross-trait assortative mating. If you add in even a small amount of misdiagnosis, say 5%, and you go out another generation or 2 of assortative mating, you can fully explain the genetic correlations for some of these traits with cross-trait, cross-mate, assortative mating. Another area where people are using these polygenic estimators is to identify the cell types or tissues or periods of development that underlie these components of shared genetics. And here the problem gets much, much worse. So these different colors denote different types of overlapping enrichment. And this purple one is the one where there's absolutely no overlapping enrichment. Say that the truth is that the enrichment for height is in some kind of open chromatin in bone precursor cells or something like this, and the one for education is in some kind of neuronal phenotype. Because there's no overlapping enrichment, all of the false positives that come back from this bias driven by assortative mating are going to be exactly in line with the wrong trait. And so the enrichment that you see is nearly perfect in those regions. And so it looks like the genetic correlation specifically in those regions that are unique to one trait and not the other will seem the most correlated. So since then, we've expanded to sort of more complex mating regimes. The first one I want to talk about is a more high-dimensional mating. Now, there are oddly, I mean, if you think about it, it's odd, but you know, whatever the papers got written, sort of proposing, there's probably 1 or 2 latent factors that all of humanity is using to make their spouse. So, you know, I mean, we felt a little silly putting this in because it seems, but you know, the papers are there and we got asked to do it. So here's the canonical correlation analysis over some traits in the UK Biobank, and unsurprisingly, you know, spousal correlation requires more than 2 latent components to sort of capture all of the variation. And if you go back and you rerun these simulations looking at multi-way cross-assortative mating, where you have, in this case, 6-variant assortative mating under these different psychiatric conditions, the problem gets much worse. It further inflates the association. So even without a misdiagnosis rate, for some of these pairs like ADHD and bipolar, and alcoholism and schizophrenia, you can fully explain genetic correlations between these traits using cross-trait assortative mating. Now, that's not to say that there isn't really truly there. I'm just saying in the simulated framework, we can get all the way there. And I'd say a reasonable simulated framework. Another thing we looked at in the same simulation setting was how many of the associations that you get back are going to be so-called off-target. That is, they're for the other trait. They're not for the trait that you're studying as you start increasing your sample size. And so for the GWAS that we're doing, up to, maybe this is UK Biobank over here, it's unlikely that, even for traits that are highly assortatively mated on, that many of the variants that we're detecting are for traits that are just assortatively mated with height on. But I think the last height mega GWAS was like 4.5 million. Our prediction over here is that, all of the juice is in all of the discoveries are going to come from variants that are really not at all height variants, that they're just variants that are associated with other traits that we're cross-assortatively mating on. I want to talk about now one extension that was motivated by some other uses of genetics outside of genomic medicine. So you might have seen some of these articles talking about policy for education on the basis of your polygenic risk scores, which I think have a whole other host of ethical problems. But let's do a thought experiment here. We're going to have one trait, height, that's genetic. Its heritability is going to be 60%. And we're going to have two other heights. We'll call one wealth. This is a cartoon. This isn't based on anything, but I think it's pretty reasonable. Okay, we have two other traits. They're called wealth and educational attainment. And in this cartoon example, neither one of them is genetic, or there's epsilon genetics there. Okay, and if we start at generation 0 and we do a GWAS, or we estimate heritability or genetic correlation, everything works out just like all of the estimators say they should. The heritability of height is 60%. We get a bunch of GWAS hits. The heritability of wealth and educational attainment are 0. Genetic correlations are 0. There's nothing there. And now we're going to assortatively mate, like we said before, between educational attainment and height. And we're going to do something which is, probably unbelievable, which is suggest that people who have a lot of money pay for their kids to go to college. Okay, and we're going to march forward in time. And what happens is that all of a sudden wealth looks genetic, education looks genetic, education and height become genetically correlated, wealth and education become genetically correlated. And so some of these social science papers that are talking about sort of a genetic basis, self-determination, and I was born with this, it's in my DNA, are susceptible to this kind of bias. So I really liked this cartoon. Similarly, this type of vertical transmission exacerbates further. So if we think about it, this is genetic correlation between, say, height and educational attainment. Here is just vertical transmission by itself. Here is just cross-assortative mating by itself. And when you put them both together, you get a quite large increase in the estimates of these parameters. Some recent work from a number of groups has begun exploring a return to not GWAS of unrelated individuals, but what could be done looking with siblings and trios and really transmitted alleles. And for a number, but not all, of the types of simulation we investigated, those do have the properties that the authors claim. They land on the direct effects, albeit with quite a lot more noise, and so the studies would have to be quite larger. The areas that we found where they tended to fail or were problematic were in ascertained studies. Oftentimes you do these trio studies where you require that one of the siblings has autism or one of the siblings has, disease X of interest, or you notice that in UK Biobank the ascertainment is not completely at random, that people tend to be a little bit wealthier, a little bit healthier, and so on. And this can induce bias in those, at least in the sibling form of the study that we looked at. Okay, I'm going to end on some newer directions. So we were a small part of and enjoyed this paper from my colleague Chun Fan, where he was looking at correlations of spousal pairs across generations and around the world. They largely recapitulate one another. That is, the spousal correlations of psychiatric diseases tend to be the same in Taiwan as they are in Denmark, as they are in Sweden, and as they are in the United Kingdom. But there was an interesting bit that he looked at in this paper, which was the prediction of the spousal correlation onto the parent-offspring correlation. So that is, if my spouse and I are correlated on some trait, how does that trait then project onto the next generation? And this is related to some work from Naomi Ray's group that she published in JAMA Psychiatry, which is sort of thinking about the changes in prevalence of these diseases as a function of these mating patterns. So if people with proclivity for a disease tend to mate, will it increase the prevalence of the disease over time if you start off at random mating? And these are a function of heritability and the strength of the assortment and so on. We started thinking about this in relationship to some questions that are maybe more classical, and in particular with respect to sexually antagonistic selection, which is, does a polygenic risk for disease, even if you don't have the disease, relate to the number of kids that you're going to have? And so there are similar studies that have been done from Cowrie. I think there's one from Peter Vischer also, and we've been sort of expanding into larger populations and sort of revisiting these questions. And we found a couple instances. So height is the classic one. A couple people have found this, and so we include it as a positive control. If you are male and you're taller, it's associated with having more kids. But if you're female, having a genetic proclivity for being shorter results in more children. Another one of these interactions we saw was an autism spectrum disorder, where men who are not diagnosed with autism but have a higher polygenic risk score for autism tend to have fewer kids than women who are not diagnosed with autism who have more children. That is, if you have a higher genetic predisposition for autism and you're female but you're not diagnosed, in UK Biobank you have slightly more children than if you have a lower polygenic risk score for autism. Okay, I'm going to stop there. This is Richard back here, and this is Jamie. And I didn't find a picture of Morton. Thank you.